Imagine that your body is a kingdom of cells, each with specific duties that keep the kingdom running smoothly. If the kingdom is ever threatened by invading pathogens, the immune cells act as the kingsmen, sworn to defend the kingdom. Often, they're successful and the kingdom returns to homeostasis, but in some cases, they overreact and in their bloodlust begin decimating the kingdom itself. The term sepsis refers to this extreme, self-destructive immune response. Typically, sepsis involves simultaneous hyperinflammation or immune activation and immune suppression, a complete dysregulation of immunity. The contradictory signals lead to a disease so varied that in the future, scientists expect sepsis will be not one disease, but several related ones. In general, though, sepsis begins with inflammation, meaning fever, rash, and swelling. Then, it becomes severe sepsis, as the overzealous immune response begins causing organ damage. Septic shock is the most advanced stage, at which multiple organs are failing as the acute immune response travels through the bloodstream, causing blood pressure to plummet as the integrity of the blood vessels is compromised. The Kingsmen destroy the roads as they move from town to town, organ to organ. Though any infection at all can cause sepsis, infections of the gastrointestinal and respiratory systems are the most common. COVID-19 deaths, for example, are believed to be due primarily to septic shock. Anyone can get sepsis from an infection, though sepsis itself is not contagious, but those with naturally weaker immune systems are especially susceptible to it. Sepsis deaths can represent about 20% of yearly deaths worldwide, and although full recovery is possible, many sepsis survivors experience chronic symptoms and eventually die themselves. The better we understand sepsis, the more we can reduce its mortality rate. So let's start with how the Kingsmen turn against the kingdom in the first place, how a homeostatic immune system becomes imbalanced.
In a kingdom of immune defense, unseen enemies threaten its harmony, but defenders are armed with the knowledge and recognition tools to thwart them. Pattern recognition receptors detect invaders, signaling alarms and allowing molecular guardians to enhance defenses. Soluble shields like chemokines and complement proteins fortify borders against persistent threats. Vascular gatekeepers regulate blood vessel leakiness, allowing swift movement of immune effectors. Adaptive architects summon T and B cells, bridging innate and adaptive defenses for resilience against future threats. Commensal bacteria further regulate the response, aiding in the production of soluble shields. Through the chaos and challenge, the kingdom remains united in its defenses. However, disruptions to the gut weakens the barriers. When our gut microbiota is disturbed, it weakens the barrier, allowing harmful bacteria to spread to distant organs. This disruption affects immune responses and can lead to reduced defenses against infections in the bone marrow and lungs. Certain bacteria in the gut help regulate immune responses, but when the microbiome is disturbed, this regulation is thrown off. Additionally, gut-derived substances affect the lungs' immunity, so disruption increases susceptibility to respiratory infections. The body's responses to this disruption includes the release of signals that keep the immune system in overdrive, fueling inflammation and immune dysfunction. This cycle, along with the activation of immune receptors, worsens with sepsis by maintaining inflammation and immune suppression. Persistent immune activation can lead to a condition called PICS, characterized by ongoing inflammation, weakened defenses, and catabolism. This condition reflects the long-term consequences of sepsis, where the body's recovery efforts are hindered. Overall, the journey through sepsis involves a delicate balance between inflammatory and anti-inflammatory responses, determining the outcome from battling pathogens to long-term health impacts.
So now you may be wondering, what causes sepsis? What's its main mechanism? Unfortunately, the pathogenesis of sepsis has not yet been fully understood. I want to explain the relationship between cytokine storm and sepsis first. It is good that your immune system detects the pathogens and the cytokines guide your immune cells to the infected regions and thus activate more cytokines. However, if this feedback is out of control, then too many activated immune cells will accumulate and cause organ damage and failure, leading to shock and possible death. This uncontrolled activity of pro-inflammatory cytokines is considered to contribute to injury in sepsis. The next question is, what cells actually contribute to sepsis? Neutrophils can contribute to hyperinflammation in sepsis through the release of proteases and reactive oxygen species. They can release NET to trap acute bacteria. However, excessive NETosis can induce intravascular thrombosis and multiple organ failure. The imbalance in the release of pro-inflammatory or anti-inflammatory factors is a central link in the pathogenesis of sepsis. In addition, the endothelial and epithelial cells can be damaged directly. This will lead us to the relationship between sepsis and coagulation and complement systems. Coagulation activation and anticoagulation impairment and fibrinolytic system are hallmarks of sepsis. Activation of the coagulation system and thrombin generation depend on the expression of tissue factor on activated monocytes and endothelial cells. Meanwhile, pro-inflammatory factors inhibit the anticoagulant system, especially the protein C system. Individual differences of sepsis patients make the study of specific genes a current hot topic. Current research suggests that certain sepsis-related specific genes or gene expression variations directly affects the host's susceptibility to sepsis, as well as response to treatment and prognosis.
In sepsis, our body's immune system faces a critical challenge, including the internal changes that suppress its ability to fight back. A big problem is the increased apoptosis in essential immune cells, including follicular dendritic cells, natural killer cells, CD4 and CD8 T cells, and B cells. For T cells, sepsis induces a state of exhaustion. This state includes increased release of anti-inflammatory cytokines like IL-4, IL-10, and IL-37, which can inhibit the release of pro-inflammatory cytokines by T cells. The remaining B cells also show signs of exhaustion, unable to effectively present antigens or produce the necessary signals for a strong immune response. Monocytes and macrophages also release IL-10, which has several immunosuppressive functions, as we've just seen. Namely, it inhibits T cell proliferation. The release of pro-inflammatory cytokines by these monocytes and macrophages also activates naive CD4 T cells, which can differentiate into different T helper cell subsets. We can see how the immune system is caught in a complex balance between trying to fight off the infection, but also trying to prevent excessive damage from inflammation. The presence of myeloid-derived suppressor cells, also known as MDSCs, increases, which makes the immune suppression worse and leaves the body more susceptible to secondary infections. A network of checkpoint regulators like PD-1 further diminishes the immune system's ability to fight. So while regulatory T cells and MDSCs create an immune suppressive environment interfering with the functions of T cells, dendritic cells, macrophages, and PD-1 help in maintaining the suppression. Finally, reprogramming of immune cells leads to a reduced capacity for a pro-inflammatory response, a state known as LPS tolerance. This state significantly disrupts the body's ability to mount an effective defense.
As the battle rages on and the enemy becomes more adept at breaching the kingdom's defenses and causing an overreaction by the Kingsmen, the medics of the kingdom scramble to come up with an antidote to the Kingsmen's madness. By identifying and grouping the behavior and individual mechanisms contributing to the Kingsmen's madness, they can better treat each individual using specific antidotes. Sepsis heterogeneity can be characterized into endotypes, which are biological subtypes based on pathophysiological mechanisms, or subphenotypes, which group together a certain set of features that allow for therapies to be more specific to the complexity and variation between sepsis patients. For precise medicine, prognostic or predictive enrichment strategies are used to help select the patients with a high chance for sepsis-based mortality, or their madness, or based on their response to specific antidotes respectively. A group of the kingdom's medics begin to classify which type of sepsis spell was casted, based on host response biomarkers such as temperature trajectory, type of organ dysfunction, or other clinical markers. The medics are able to conduct trials based off certain subphenotypes, such as providing anti-TNF antibodies to those with IL-6 to reduce severe systemic inflammation. The kingdom's resident inventor has advanced their omics technology. Now the medics can analyze the proteomic metabolic data of the affected to predict mortality and how the spells use a combination of genetics, metabolites, and cytokine levels to lead to sepsis pathophysiology. Transcriptome data of blood leukocytes is also used to help group those under the sepsis spell, as certain antidotes such as corticosteroid antidotes can be fatal to certain endotypes. Even with the advancement of the kingdom scientists in omics, immune profiling technology, the medic's analysis and antidotes based on tissue response, immunological profile, and clinical phenotype, the sepsis spell targets too many different pathways in different individuals, leaving the kingdom with a better understanding of the spell, yet few effective treatments. So, is it over for the kingdom? A new emerging area of artificial intelligence, or AI, is currently being developed to better spot patterns, group, diagnose, and provide prognostic to those affected. This leads to the King's Declaration of Sepsis 4.0, being an identifiable heterogeneous disease that must be treated with patient-specific medicine, while also directing more scientists to focus on future research into post-sepsis immunity to lead to a more stable, better quality of life for the kingdom post-sepsis.